Hello, my name is Mark van Loosdrecht. This module of the Environmental Biotechnology MOOC deals with the elemental cycles and the crucial role of microorganisms in the circularity of the natural ecosystem. The first element we will discuss is carbon, the major element for life on Earth. Photosynthetic organisms, plants and algae, use solar energy to convert atmospheric CO2 into organic carbon. This is known as autotrophic carbon fixation. Roughly half of the global carbon fixation is performed by algae and the other half by green plants. The organic carbon produced supports not only plant life, but is essentially for all visible life forms, including animals and insects, which depends on plants for food. Over geological timescales, part of this organic carbon can be stored as coal or crude oil. While carbon dioxide can also be stored in the Earth's crust or oceans in the form of carbonate minerals. Microorganisms convert organic carbon back into CO2, preventing the accumulation of dead biomass and ensuring a continuous supply of carbon dioxide for new photosynthetic activity. This decomposition process also releases essential nutrients for the growth of biomass, making them available for new life. When oxygen is absent, the degradation of organic matter proceeds through anaerobic conversions, including fermentation and anaerobic respiration, ultimately leading to methanogenesis, the formation of methane. Methane usually escapes into aerobic environments, where it's further oxidized to carbon dioxide and closing the cycle. In this lecture, we will focus specifically on aerobic conversions, the microbial processes that oxidize organic carbon and methane to carbon dioxide in the presence of oxygen. The main function of aerobic microbial growth is to close the elemental cycles of carbon, nitrogen and phosphorus, as these elements are released during the complete oxidation of organic matter to carbon dioxide. We distinguish two main forms of aerobic microbial growth. First, heterotrophic growth. This is growth on organic substrates, with oxygen serving as the electron acceptor. Secondly, autotrophic growth, growth of inorganic substrates such as ammonium, sulfide or ferrous iron, where oxygen acts as the electron acceptor. This conversion leads to the formation of oxidized products like nitrate, sulfate or ferric iron. Under aerobic conditions, all the energy contained in the substrate can be harvested for microbial growth. The substrate is fully oxidized, like in a fire, releasing energy that microorganisms capture for biomass production. The oxidation of organic carbon, the so-called catabolic reaction, produces carbon dioxide, water and energy. This energy is then used in the anabolic reactions that build new cellular material, a complex biochemical process beyond the scope of this lecture. In brief, organic substrates such as glucose are gradually oxidized to carbon dioxide. The electrons released during this oxidation are transferred to an intracellular electron carrier called NADH. These electrons are subsequently passed to oxygen through a series of reactions that generate cellular energy in the form of ATP. For more detail, please refer to standard biochemistry textbooks. In aerobic microbial growth, oxygen serves mainly as the terminal electron acceptor. The amount of energy produced in this process is therefore proportional to the number of electrons transferred to oxygen or the oxygen consumption. When we compare aerobic microorganisms in terms of growth yield per unit of oxygen consumed, we find that their yields are remarkably similar. Contrasting, when growth yield is expressed per unit of substrate consumed, there is a significant variation in yield values. Some highly reduced compounds, such as methane, ammonium or benzene, always needs first a direct reaction with oxygen. In such a first metabolic step, the available energy is released as heat in the initial oxidation reaction, rather than being captured by the cell to sustain growth. For instance, during methane oxidation to CO2, 8 electrons are released in total, but 2 are directly used in the first conversion of methane to methanol with oxygen. Thus only 6 electrons contribute to the cellular energy generation, meaning that only about 75% of the total energy in methane can be utilized by the cell for growth. Microorganisms can also use alternative electron acceptors, topics that will be covered in other lectures. The most common example is denitrification, where nitrate replaces oxygen as the electron acceptor. From a microbial standpoint, aerobic and denitrifying growths are almost identical except for the electron acceptor used. All bacteria capable of nitrate respiration can also use oxygen, while many, but not all, aerobic bacteria can use nitrate in the absence of oxygen. When oxygen is the acceptor, it is reduced to water. When nitrate is used, it is reduced to nitrogen gas, so it's a nitrogen atom which changes in valency or takes the electrons. One of the main applications of aerobic microbial growth is in the removal of organic pollutants from wastewater. If organic matter is discharged into natural waters without any treatment, bacteria will consume the available oxygen while growing on the organic matter, leading to oxygen depletion. This results in anaerobic conditions, causing unpleasant odors, black water, and deaths of aquatic life. The amount of organic matter is generally expressed as chemical oxygen demand. That is the amount of oxygen required to fully oxidize all organic carbon to CO2. For example, complete oxidation of 1 millimole per liter of glucose requires 6 millimole per liter of oxygen. This corresponds to a COD of approximately 192 milligram per liter, 6 times 32. Municipal wastewater typically contains between 200 and 600 milligram per liter of COD, making it relatively dilute compared to industrial effluents. Aerobic treatment processes are highly effective and widely used for pollutant removal. However, certain compounds, particularly strongly halogenated substances, like polychlorinated, brominated or polyfluorinated compounds, are resistant to aerobic degradation. It's difficult to oxidize them. And they may require anaerobic treatment because they are easier to reduce. This will be discussed in later lectures. Aerobic microorganisms generally grow rapidly and produce a large amount of biomass because they can use the full energy available in the substrates they use for growth. However, aerobic processes have two main drawbacks. First, oxygen transfer limitation. Oxygen is effectively purely soluble in water, only around 10 milligram per liter maximum in ambient temperatures. Therefore, a significant amount of energy is needed for aeration to bring the oxygen into the water. And secondly, the high microbial yield leads to large amount of excess biomass, which is typically considered a waste. In most Western European countries, it needs to be incinerating, adding to the operational cost of wastewater treatment. Thus, while aerobic processes are very efficient in removing pollutants, their energy and sludge handling requirements motivate the search for alternative or complementary processes, such as anaerobic treatment. In summary, aerobic processes are used most widely in environmental biotechnology for wastewater treatment and soil or groundwater bioremediation because of their efficiency in removing all the organic carbon. The main engineering challenge lies in the oxygen transfer due to its low solubility. The final product of aerobic oxidation is carbon dioxide, which, unlike methane from anaerobic processes, has no real reuse potential. Nonetheless, aerobic systems remain essential for maintaining a clean and safe environment and clean drinking water in the end.